Okay, so this is my final video. I still haven't gotten used to taking videos of myself in this class this semester, but here it is, and I guess I will just start with the first question, which is, what was your favorite topic and why? Explain why. So I'm going to skip how it feels or what feels applicable to my future career because that part didn't really interest me as much this semester. What most interested me was learning everything that we learned in Module 7 of the applications of microbes. You would think that going into nursing, my interest would have been like the biology and the morphology of like microbial organisms, but honestly, there's such a difference between the biology and morphology of microbial organisms compared to like human cells in the human body. Like it's very different, and I found this more interesting because talking about all of the different ways they interact with us and help us and benefit us, and how they interact with the environment, it just reminds me of the way our body system works together and interacts together. One thing I was not interested in learning about in this unit was the food microbiology. Not because it's not fascinating, but I worked in food before. My milk is my milk, my cheese is my cheese. I'm not so much interested in which microbes make that happen and why. I don't know, it's just not my cup of tea. But other things like the way microbes communicate with each other in their environment and the competition between microbes, that sort of brought me back to like human anatomy and physiology, the way that our cells talk to each other and interact with each other in order to carry out different actions or different defenses. And our body cells obviously don't compete with each other for like in survival of the fittest fashion the way that microscopic organisms might in an environment compete for survival, killing off, other species of themselves or whatever. Obviously, our cells in our body don't do that. They more or less work together to make the whole unit work. But it just kind of brought me back to how they interact with each other and how they can talk to each other and work together to do different things. Another thing that I found really interesting in this unit was when you were talking about the metagenomics. Kind of reminds me of like lab work, right? So you're going to take a sample of your environment and send it to a lab and essentially have it evaluated to see what different microorganisms are living in that sample that are potentially in that environment, how many, and just all kinds of different information about them. And even if there are some unrecognized because we haven't done DNA sequencing on them, that's a way going forward into the future to discover new DNA, new microorganisms that we aren't aware of yet, which to me is, I feel, similar to like getting blood work done. Like you're testing for certain things, the presence of this or that. And I don't know, I think that's kind of correlated to my future career, and that's just kind of fascinating to me. I also really liked talking about the biogeochemical cycling because so many of those cycles, those are things that no one ever thinks about in their everyday life. We, especially like the carbon cycle, we hear constantly about carbon emissions, oh, the environment, oh no. Okay, great. We do as a planet, I think, look at our carbon emissions, okay? But then you get into all of the carbon emissions in this class. Like, you're not thinking about the fact that this heterotroph is eating this chemotroph, and this heterotroph is eating this heterotroph, and it just goes and goes and goes, and every single time something moves through the food chain, carbon is being emitted. Like, who thinks about that? And if it wasn't for these microorganisms that are able to use that carbon in their own metabolic processes, in order to produce something that we can use and get rid of that carbon for us, that's awesome. Who thinks about that? So, my dog. Module 7 in the application of microbes, I could just talk about all the other stuff, but I'm obviously going to answer the other questions related to that. I think was just really the most interesting for me. It was most fun to take the notes and study all of that information. So for question 2, if you had an unknown microbe, what steps would you take to determine what type of microbe, fungi, bacteria, virus it is? Are there particular characteristics you would search for and explain? So yes, the first thing that you need is obviously a microscope, and the most common is a compound light microscope. So you're going to need your sample and your microscope. You're going to first need to determine whether it's a prokaryote, which is going to cover the bacteria and archaea, or whether it's a eukaryote, which is everything else. In order to determine whether if it's a prokaryote, you need to first look for the shapes. So prokaryotes come in circular form, spiral form, or circular or spherical, as well as like tubular form. Tubular rods, I think is what you called it in the lecture. So you can first identify by a shape and then kind of go off of that. And then from there, you're going to need to separate whether it's a bacteria or whether it's an archaea. So a bacteria, you're going to be able to stain. So the bacteria is the cell wall is made of peptidoglycan, where the archaea is not. So that allows you to use different stains, like your positive or negative stains, and all the different types of stains that you can use, whether it's like a positive, negative, because of the negative or negatively charged peptidoglycan, or I hate getting tongue-tied on my words. So whether it's a differential stain or a simple stain, you'll be able to see more about if it's a bacteria or an archaea. I'm not sure if I said this already, but I want to make sure that I cover that a prokaryote has no nucleus and no organelles, and a eukaryote does. It's just a bunch of genetic information, ribosomes for reproduction inside of a capsid. So if it's not a prokaryote, then it's a eukaryote, which is everything else. So eukaryote is going to have a nucleus, nucleolus, and organelles. As for the next category, which is going to be the non-living, the viroids, the viruses, and the prions, I think those are all going to be pretty simple to identify under a microscope, because a virus is just a capsid with the genetic information inside. It might have proteins present, and it needs a host for replication. So, and then a viroid is going to be just a single piece of RNA replication material, and it's going to obviously stand out, I would think, right? Like, it's going to stand alone because it's just a single strand of RNA, or a single piece of RNA, to just rebuild itself. And I'm pretty sure that you said it seeks plants. Usually it tries to reproduce in plants. And then prion is going to be our own genetic information. It's going to be our own protein, not genetic information. It's going to be our own protein that was just folded wrong. It's just going to be a messed up protein. So those three things I feel like might be pretty simple to identify with a microscope. And then as for the fungi, and so the fungi, I think all you said was that they range from a single-celled yeast all the way to, oh no, what did you call it? It's not microscopic anymore. All the way to a growing fungi. So I'm not exactly sure how you would identify that under a microscope, but and then anyway, with the protists, which are going to be your algae and your protozoa, I also feel like those might be pretty simple to identify because the algae is going to be a chemotroph. And I feel like, according to the picture that was on the slideshow, you're going to be able to tell that there is like the pigments of like photosynthesis occurring. Did I say chemotroph or phototroph? I meant phototroph. So your algae is a phototroph, and so I feel like you're going to be able to see that. And then the protists or the protozoa is just a chemotroph. It's so you'll be able to tell the difference between a phototroph and a chemotroph based on cellular differences. You also did say that protists can also be identified with their short cilia. So question number 3 is probably my favorite because I am fascinated by how the human body works. It literally makes me want to cry sometimes. It's amazing. So imagine you're interacting with someone who does not think that vaccines are effective, and use your knowledge to explain how you would have a conversation with them. So you would first have to very surface level explain how the immune system works, right? You start with your, you have your innate immunity, which is what we're born with. It's all of our natural defenses to keep ourselves well and to keep bacteria and viruses out and rid the body of them, or actually mostly prevent them from entering the body at all, but then eventually rid the body of them. And when that fails, then our adaptive immunity kicks in, and that is where cells really go to work. So you have to kind of explain the B cells and the T cells. Your humoral immunity is where your B cells come in, and that is, they're outside. They're extracellular, and they're floating around, and they're looking for foreign bodies, pathogens that shouldn't be there. When they are found, those B cells change. They recognize the pathogen, and oh my gosh, I can't believe I'm drawing a blank right now. So they recognize the pathogen, and then so those B cells, two different things happen. They transform into memory B cells, which means, okay, they recognize this pathogen, and those memory B cells are going to remember that pathogen next time it's encountered. And they're going to be ready to signal the body, hey, this is bad. We don't want this here. And I remember this pathogen, and we're going to deal with him the way we dealt with him last time. And then they're also going to become, oh, goodness gracious. Okay, so the other B cell becomes the cell that creates the antigens to get rid of those antibodies. So you've got one part that's going to remember, hey, this is bad, and the other part that says, oh, I got the antibodies to deal with that. Let's go do that real quick. So when that system fails and a pathogen is able to enter a cell, B cells cannot follow it. So that's where T cells come in. That cell of the body says, hey, I'm compromised. I'm invaded. You gotta, I need help. I need sacrificing itself, basically. So the T cells come and get rid of that whole cell and everything that was a part of it, including the pathogen. When you get sick and your body, so B cells are part of your humoral immunity, and then the T cells are part of that cell-mediated immunity when they come and just get rid of the cell altogether. When those two things don't work and you end up sick, let's just say you get sick and then you get well. So your body now has an immunity to that sickness for a while, because while you were sick, your body was creating antigens to fight this pathogen or these antibodies. And essentially, that's exactly what a vaccine does. A vaccine is a fake version of a virus or a pathogen. It's a deactivated or dead version of that virus that's either not going to have the ability to replicate, or it's going to be a much less it's not live or active, and it's going to allow your body to see or to encounter that pathogen and say, oh, we need to create the antibodies for this so that we're ready when we encounter the real thing. So basically, it's the same thing as if you get sick and get well, and then your body's going to be able to fight off that pathogen for whatever amount of time on its own, because you've developed that in your immune system. The vaccine is tricking your body into thinking that you encountered this virus, and now your own immune system is going to build protection for it's not the vaccine that's protecting you. It's your own immune system that's protecting you. The vaccine is just tricking your body into protecting itself better. So for question 4, describe 2 positive and 2 negative relationships humans have with microbes. What are some advantages and disadvantages? So one of the bad relationships with microbes that always comes to my mind first when we're talking about something like this is Staphylococcus. It's very interesting to me that Staphylococcus is part of our normal human skin microbiota. Like, I had no idea that Staphylococcus was just around everywhere all the time on us. Thought it was something that you never wanted to encounter, never wanted to keep that away, but turns out it's all over the place. Unfortunately, because of that, there's always a potential risk for some kind of Staphylococcus infection. Like in the PowerPoint, we went over scalded skin syndrome, as well as like a hair follicle infection and impetigo, and I know there was maybe a couple of others with certain strains of Staphylococcus aureus, or whatever else. I know there's certain strains of that can cause these really bad things. So unfortunately, it's part of our normal, microbiota, but therefore it's always a potential risk that the strains might come into play and we might have negative consequences from them. Some good things about our relationships, the relationship between humans and microbes. I liked how we went over the conversion of elements. Microbes, they're our little lifesavers, I feel like, because of all the bad things that we put out into the environment. Like I know we went over oil spills and, the carbon cycle, nitrogen, all these different things that are just constantly bad things that we're constantly putting out into the environment. Trash, oil, and whatever else, organic materials. They do so much work to convert those materials for their own use, and then the byproduct of that is usually something good for us that we need, or the byproduct or the end result of them taking all of our bad things and converting them to something that we need, like not CO2, like O2 or H2O, amongst other things. That's definitely a very positive relationship I feel like we have with them. I feel like through all the evolution, like we talked about closer to the beginning of class, through all the evolution, how we've evolved together, we help them by supplying them with what they need, and then they help us by supplying us with what we need. So when we talked about mycorrhizas, even though that's more of a relationship between a plant and a microbe, that also benefits us because that plant benefits us. I think it's really cool how it's like it's a chain reaction, because the plant provides this microbe with a home, the microbe provides the plant with nitrogen, and the plant provides us with nutrition. So in this chain of events, we in turn have a relationship with microbes like that, or even like hyphae that do the same thing. They help these plants grow, and then the plants in turn help us because we eat them and we use them. Okay, I'm already past my 20-minute limit, so I'm going to try to go through this fast, but still be detailed. So many applications of microbes exist. List and describe 5 uses of microbes. So the water treatment would be like what I would consider the greatest. I knew water treatment plants existed. I knew they clean the water, blah, blah, blah. Does anyone really know how that's done? Nope, I didn't. So come to find out, we purposefully, specifically utilize microorganisms in order to clean our water for us and convert it back to H2O. So especially in like secondary water treatment, and using the BOD level to, who would ever think that too much oxygen in water could be a bad thing? And then you find out that it's an indication that there's too much biodegradation going on, which means there's too much bad stuff in the water to drink it. Like, that's crazy. So we use it for water treatment. We also depend on microbial life in landfills. Like, a landfill is something else. Who knows how that works? It's a giant hole with a bunch of trash. So they specifically layer it and fill it with at certain levels with certain things, and they depend on the microbial life present to degrade all of our trash and all of our things, all of our junk and our waste. We're dependent on those microbes in order to maintain landfills. Food. Food is another huge one. So yeast for breads, fermentation of milk by Streptococcus and whatever the other ones are, I can't remember at the moment. Fermentation to produce yogurts and cheese. And then molds, also. Molds for different kinds of cheese. We depend on that as well. Fermentation of alcohol is like huge in our society, huge in our culture. It's global. Alcohol is global. That requires microbes. Mushrooms. We eat mushrooms. We're literally eating a microorganism, same like with bread and yeast, even though mushrooms aren't really a microorganism, but you know. That's all I got. That's my 5 uses of microbes. So This class was a hard one for me. It's so my the micro world is so different than like what the human body is. Like these microbial organisms are so different than, I don't know. It just wasn't what I was expecting, but it definitely was an eye opener in this course to think about the way that I see microbes everywhere now. Like literally every single day, I tell people, oh, you know why that happens? Microbes. You know why that happens? Microbes. Like I see them everywhere and like I understand it better now. So I hope that this was good and thank you for sharing all your knowledge in this course.